that always looks good. That always looks good with NIGMS.
Oh, yeah. Oh, yeah.
So congrats on getting the competitive renewal.
Yep, we're pretty excited. A lot of work, but that's okay. We're working with Joan Lappy's working on our professional development board. Do you know Joan?
Right.
So we're pretty excited. How's your Cobra?
Well, we've just done our PPR, so we're recovering from that now that we know how much work it is. Uh, um, so I think year two is going to be consolidation phase and get everything up and going. Most of the major things are up and going, and now we just got to make sure that we can sustain it. And
Yep.
hopefully we'll have a bit of time now to, get our bearings in order to do that.
Yeah, it
Um,
kind of goes on, doesn't it? Heather knows I don't like those RPPR reports.
well, I'm hoping that the admin team will take care of that going forward, and they just come back to me for high level stuff, or what I need to do as the PI of a particular component. So
Well, and if you're tracking an evaluation groups going, that, a lot of that should just be secondhand for them to pull that up. I know that's
that's
part of what we're working on, right?
great, because we would love to learn from your experience there. We're going to start on that this month and next month, and trying to figure out what we can do with the data that we are collecting already. And so, uh, yeah,
Good.
so that we can continue to improve and evolve.
Yep. Well, listen, I think it's 10:01. I think we're going to go ahead and I'm going to introduce Dr. Peter Steyger, who he's had 3 decades of experience investigating the mechanisms of hearing loss due to hospital prescribed medications. He was recruited to Creighton in 2019. He now leads the Pan Omaha Translational Hearing Center to develop new strategies to preserve or restore hearing from congenital or acquired forms of hearing loss. And this is that the title of your new Cobra?
That is indeed,
Okay.
yes.
And they just received your Cobra funding came last spring, was it, Peter?
That's correct. That's
Okay.
correct.
And we're doing some collaboration, as people know, in our idea CTR. Creighton is one of our partners. So we are obviously doing a collaboration with Creighton and are very interested to hear about your new translational hearing center. So without any further ado, Dr. Steyger, welcome. And please go ahead with your presentation.
Okay. Thank you so much, Dr. Zimmerman, for that gracious introduction. And yes, this is an introduction to our CTR, to our COBRE, the Translational Hearing Center, and there will be some resources that will be available here to the UNMC community to participate in this center, and also to those who are joining us from Boys Town National Research Hospital as well. Um, so I will begin. Um, so, so the center was established in 2019 when I was recruited by Jian Zhou, who we can see here in the upper left of this image, that was designed by Creighton to celebrate the initiation of the Translational Hearing Center, and it was a vision of Dr. Zhou, and we were initially funded by the Bellucci de Paoli Foundation, which was set up by this fellow here, Richard Bellucci, who is a graduate of the Creighton Medical School in 1942, and he went on to become an otologist in chair of several ENT departments in New York City. For those of you that are familiar with otologic surgery, He did a lot of middle ear surgeries, and he developed a pair of scissors that had a very fine blade, so that they would enhance the effectiveness of these surgeries. And they became known as the Bellucci scissors, and they became the financial foundation uh, for his philanthropic efforts. So, so that was how the Translational Hearing Center began, and the overall goal is to build a critical mass of center-affiliated investigators that identify strategies and candidate compounds that are called ototherapeutics to preserve or restore hearing and vestibular function. And I'm mainly going to talk about hearing, but any reference to hearing is also going to include vestibular function because both sensory organs are contained in the same bone structure called the inner ear. And the rationale for focusing on these two particular sensory modalities is that hearing loss affects at least 500 million people worldwide. And noise-induced hearing loss affects millions each year, and they can be from military, occupational, or recreational etiologies. And there are more than 400 genomic and mitochondrial polymorphisms that lead to hearing loss. And there are also many iatrogenic causes of hearing loss. That is, hearing loss that originate within individuals that are in hospitals, and they're usually medication based, typically from aminoglycosides or cisplatin, or combinations of drugs such as loop diuretics with other non-ototoxic drugs, but when they are administered together, they cause hearing loss. And then there are additional causes of hearing loss that we are looking to get involved with as we continue to grow and expand over the next few years. And these include age-related hearing loss, teratogenic induced hearing loss, and these include alcohols and solvents. And then there's tinnitus and central auditory processing disorders. And the consequences of these forms of hearing loss tend to be uh, tend to be quite common across all these different forms of etiologies. And if they're prelingual, they have a cost of $1.5 million over the lifetime of a single individual, and has a socioeconomic cost, and they diminishes relationships, whether they are career, familial, or friendships. Those relationships are harmed, and people do not typically reach their potential or career trajectories that they could do if they didn't have hearing loss. And in older individuals, hearing loss can accelerate aging. They damage relationships, they degrade them. Individuals can lose their balance and fall. And each of these outcomes uh, is known to be associated with cognitive decline. So our sense of hearing and balance is extremely important for a continued quality of life. So there are many strategies to rehabilitate these sensory deficits, but they all are limited. For hearing loss, we have hearing aids. They can be very effective if fitted well, but they are limited to the frequencies of surviving sensory cells. And those are mainly low frequency or, bass, low tones, and you do not hear the higher frequencies, such as those of children or cats or birds. And so again, you're missing out on wonderful auditory stimulation around you. Hearing aids also amplifies noise, and it has poor frequency discrimination, and accessibility is limited by cost and the need for good fitting to the hearing loss profile that one might have. And there's still today even a lot of social stigma in wearing hearing aids, particularly in the older generations. And it's well known that maybe only a third or 40% of people, um, or of all the people who are eligible to wear hearing aids, maybe only 25, 40% of people will wear them. So there's an underutilization because of social stigma. There are also cochlear implants or bone anchored implants. Now, these can be outstandingly effective when used, but they're invasive, it needs surgery, and there's no guarantee they will actually work ahead of time. And that's a big challenge that's facing the implementation of these strategies. And they're limited by battery life and also by surviving cochlear neurons. So if those cochlear neurons are not there, they can't take the signal to the brain. There are some auditory brainstem implants, but that's still very much in the translational phase in very small, limited clinical trials. And the same is true for vestibular implants. They're still at the translational stage with small clinical trials. So I just want to give you a little bit more background here because uh, individuals with severe bacterial infections or sepsis are treated urgently with life-saving glycoside biotics, and severe infections induce systemic inflammatory responses, and this can modulate the permeability of the blood-brain barrier. And this modulation of that blood-brain barrier can also occur in the blood labyrinth barrier that protects the cochlear and vestibular apparatus. And this increases the uptake of aminoglycosides, increasing the cochlear toxicity and functional hearing loss induced by aminoglycosides. Thus, the very individuals that need aminoglycoside therapy clinically are also those most at risk of drug-induced hearing loss. And yet, 95% of the non-clinical ototoxicity studies done to date are using healthy preclinical models. So there's a disconnect in the research world of how to prevent ototoxicity if they're using healthy subjects and not models in which preclinical models of the medical settings in which these drugs will be used. Thus, if we're going to develop candidate ototherapeutics, these must also be efficacious in the models of the medical setting in which they are to be employed. And so what we plan to do here in the center is to develop research that will identify therapeutics that preserve or restore hearing and vestibular function. And this can be both in the peripheral inner ear or centrally within the central auditory and vestibular pathways. But in order to do that, we need to have a pipeline, and this is uh, pipeline, our logical pipeline, where we can identify the genetic or clinical settings that lead to hearing loss in order to screen for candidate ototherapeutics. And then we need to identify the optimal local or systemic delivery mechanisms. And when I say local, I mean delivery locally by injection or similar mechanism into the inner ear, because the doses that are necessary to get into the inner ear can be more readily achieved if given locally. Because of that blood labyrinth barrier, it can prevent those drugs delivered systemically from getting into the inner ear. And in order to cross that barrier, you might have to give such high levels of drugs that they become systemically toxic. And so there are a number of challenges of delivering these drugs in order to reach therapeutic concentrations within the inner ear. And then, of course, once they get into the inner ear, we need to understand how they get there and their pharmacokinetic and pharmacodynamic profiles, absorption, distribution, metabolism, and elimination. And then we need to validate these hits from in silico or in vitro studies, in vivo studies, in clinically relevant scenarios in preclinical models. And then, of course, we can either go to clinical trials in humans, and this is where Boys Town at UNMC is most important for us here at Creighton, because you guys have access to the human populations and you have the expertise in doing these clinical trials, whereas we at Creighton don't have that so much. And between all of us, we hope to have some commercialization with SBIRs and STTRs, taking these to commercialization. So as I mentioned, the initial funding was the Bellucci DePaoli Family Foundation, and they provided us with about $1 million in funding over the last 2 to 3 years, which we have funded pilot projects. And of those 11 that were funded, we have 3 federally sponsored studies already. Yeah, that will bring us about $3 million to date. And we have hopefully have others in the pipeline based on those pilot projects. And the funds from this foundation has also provided infrastructure enhancements, such as purchasing a clone of a confocal microscope so that we can have additional imaging capacities. We've funded another PhD studentship, and we have a competition for postdoc and graduate student research awards to bring them into a culture of writing research proposals, because this is the nature of academic research. It's always a competitive process. And we are now in the process of negotiating a multi-year gift to continue this training paradigm that will run alongside our other federally funded efforts. And before I go on, I further want to emphasize again that this center is a tri-institutional center, which has um, uh, specific lines or boundaries of engagement. At Creighton University, we're going to be principally focusing on ototherapeutics to prevent drug or noise-induced hearing loss. And at Boys Town, with Dominic Cosgrove leading the site there, we want to focus on gene therapy to reverse congenital hearing loss. And at UNMC, with Anna Janewski, we want to focus on central neural mechanisms of hearing loss and how we can prevent that from happening. And between us all, we want to train the next generation of researchers and clinicians in these new ototherapeutic strategies to prevent or restore hearing. And then we'll commercialize these discoveries, and in doing so, become an academic leader. And the 3 locations together have collaborated to submit a proposal for a Center of Biomedical Research Excellence, or a COBRE award, from the National Institute of General Medical Sciences. And we were fortunate to have our A0, the first submission, funded earlier this spring for 5 years, and this will provide us with $1.5 million each year in direct costs. And this has been largely due to the support and interactions with other COBREs and similar idea programs here in Omaha, that include the Center for Molecular Biology of Systems, that was founded by Shelley Smith and has just completed phase 3 last year. And we were also grateful for the support of and input of Nick Sturgeon at the Center for Research in Human Movement Variability, and Laurie Leibold at Boys Town, who's leading the Center for Perception and Communication in Children. So we have two auditory Cobras here in Omaha, and these are the only two auditory Cobras in the country. So we're very fortunate in that regard. And at UNMC, we had a lot of input from Anna Janevsky with her Cognitive Neuroscience of Development in Aging Center, and the Great Plains IDEA Clinical and Translational Research Network, which really provided a lot of support, particularly in evaluations and tracking modules that were helpful for the proposal. And since that time, we have become more involved with the Nebraska IMBRE program, which was already including Creighton University because of its focus on undergraduates. And so we can synergistically support one another through all these individual IDEA programs, and we've been very grateful for that. So this COBRE for the Translational Hearing Center, we have an administrative core, and this coordinates all the activities of the center, and that includes pilot projects, funding for pilot projects, multiple research vouchers each year. We will be recruiting junior faculty, and it coordinates the mentorship of junior investigators to obtain R01 level funding. And what I mean by R01 level funding, which is the real goal for the real benchmark that we need to achieve, is a federal award that is about the value of a modular R01 of $250,000 from an NIH institute. But you can get similar funding for similar amounts or more from the DoD and from other federal sources or even the NSF. And if we can get, so that's why we're calling it R01 level funding. And by getting this R01 level funding, we can facilitate the sustainability of the center itself. Because the more funding we bring in, the more indirect costs, and the more that we can recycle the funds that we have within the COBRE to recruit new people to the area. The COBRE funding also supports two research cores, the Drug Discovery and Delivery Core. And the prime focus of this is to do screening and identifying therapeutics for the inner ear. It's, you know, the in vitro and in silico screening is not that different from more systemic applications, but the inner ear itself, because of that blood labyrinth barrier and the size of the cochlea, which is the size of a pea or a little bit bigger in a human being, there's not a lot of tissue to go around. And so in order to do PK, PD analysis, these are still very challenging concepts for the field as a whole, not just for our center. And we want to provide these services with state-of-the-art techniques to the Omaha community via a fee-for-service model, which is mandated by NIGMS. But this fee-for-service model will increase sustainability of both the core and the COBRE, not just during the life of the COBRE funding, but beyond, which is an important benchmark that NIGMS is looking for. And then we also have the Auditory and Vestibular Technologies core, which will allow not only cellular electrophysiology, but non-invasive audiometric and vestibular electrophysiological measures of function in vivo. We have advanced imaging, molecular biology, and mass spectrometry services as well, again via a fee-for-service model. But most importantly, we're going to promote the projects of 3 research project leaders at any one time, and their goal is to get R01 level funding, and we're going to provide them with internal mentors who will help them with their day-to-day and week-to-week running of projects, as well as external mentors whose primary goal is to enable these research project leaders to transition their research programs to towards translation and into human studies as they gain independent research funding. And we believe that all of these processes will enhance the research excellence and capacity to increase research funding, independent research funding, but also the potential for startup spin-offs. And I'd like to take a little bit of time to introduce several of all of our research project leaders to give you an idea of the flavor of the projects that we're most interested in. And so this is Teshbir Carr, and she's at in biomedical sciences here at Creighton. And the title of her project is Role of Macrophages and Fractalkine in Degeneration and Repair of Cochlear Synapses. And this is because noise-induced hearing loss can occur to the loss of the synapses between the cochlear sensory cells and the afferent neurons without losing the hair cells themselves. And that's called cochlear synaptopathy or hidden hearing loss. So individuals may still have their auditory thresholds, but they cannot hear or discriminate between sounds as a result of that hearing loss. And so in other systems, fractalkine, which is an immune-mediated signaling mechanism, is a positive regulator of neuron survival and synaptic repair. So she is testing the hypothesis that fractalkine can repair damaged ribbon synapses in hair cells after noise trauma. And ribbon synapses are very specialized synapses in sensory cells, not just in the ear and the vestibular system, but also in the eye. And Wally Thorson at UNMC is also studying those. And if fractalkines may be secreted by macrophages, and so she wants to know if macrophages can attenuate synaptic loss or if they can be involved in the repair of damaged synapses after noise trauma. And we're also sponsoring a research project leader at UNMC, Padmashree Raghunathan, who is in neurological sciences. And the title of her project is Effects of Prenatal Alcohol Exposure on Synaptic Plasticity and Auditory Processing. And fetal alcohol spectrum fecal alcohol is a major teratogen, and it can cause hearing loss or auditory processing disorders in up to 5% of individuals affected by fetal alcohol spectrum disorders. And there is growing evidence that neuronal plasticity is impaired in this disorder, including the reduction of the number of dendritic spines and alterations in AMPA receptor function. So her hypothesis is, does prenatal alcohol exposure impair auditory processing? And, and she's going to study this in mice, and she wants to see if this alcohol exposure impairs synaptic AMPA dynamics and synaptic transmission in the primary auditory cortex. And can we prevent this from occurring by activating TrkB receptors to normalize AMPA receptor function in a mouse model of fetal alcohol syndrome? Another project leader is Tal Teitz in pharmacology and experimental neuroscience here at Creighton. And she is repurposing a BRAF kinase inhibitor to prevent cisplatin and noise-induced hearing loss. And this is important because there are no FDA-approved therapeutics for cisplatin-induced hearing loss, or indeed even for noise-induced hearing loss at this point. And high throughput screening has identified several kinase inhibitors as cytoprotective. And one of these is BRAF kinase inhibitor, is already an FDA approved compound. And it could be repurposed as an ototherapeutic agent. And pilot data supports that hypothesis. So she's going to test if this inhibitor, defrananib, is effective against both cisplatin-induced hearing loss and noise-induced hearing loss in vivo. And more excitingly for us as a center, she graduated recently with an R01 and to expand these specific aims. So Tal, on starting this R01, has now graduated from our program. And so we have had to recruit a new research project leader, and we have successfully funded a replacement research project leader, Jeffrey North, at Creighton. And he's going to be working on the development of novel therapeutics for the treatment of mycobacterial infections. And again, there are no FDA approved therapeutics to prevent, uh, um, in this case, a minor glycoside induced hearing loss during the treatment of mycobacterial infections. And high throughput screening has revealed two compounds that can target mycobacterial proteins, not mammalian proteins. Therefore, there's negligible toxicity in mammalian cells. And if this can be translated into humans, this will reduce the dependence on minoglycosides and their non-ototoxic effects. Uh, so that could be a very, very promising development. So his specific aims are to assess whether these novel series of antimicrobacterial agents have any ototoxic or cytotoxic effects, and to design and synthesize novel selective antimicrobacterials with high potency, no to ototoxicity, and optimize physicochemical properties for use in mammalian in vivo. And so as each research project leader graduates with R01 level funding, we have another slot open, and we can then recruit off our bench of candidate research project leaders. Um, and so we will be recruiting more. And so I want to take this opportunity to describe to those of you on the call today what the eligibility requirements are to obtain this sort of funding. So any meritorious proposal will have a focus aligned with the center's mission, and this is particularly mechanisms of hearing loss or strategies that preserve or restore hearing. And there are always two slots at Creighton, and one slot alternates between Boys Town and University of Nebraska Medical Center. And as we just talked, Padma Shri is the current RPL in her second year at UNMC. And so when she graduates from the program, that slot should ideally come to Boys Town. And an RPL slot is worth $200,000 a year in direct costs for up to 3 years. And they are mentored by an internal mentor who receives 5% salary support, and also an external mentor. And the external mentors is to assist the transition of that project from preclinical studies into human studies. It doesn't mean human studies have to take place, but it's to envision the transition of that project so that it doesn't go lead on to just more and more basic science research. We want to have projects that will have a clinical impact sooner rather than later. And these individuals must be a junior investigator on a tenure track that has not yet received R01 level funding. So in other words, less than $215,000 in funds from a single federal agency. and they must submit a proposal for R01 level funding by the end of year 2 of their research project leader position. And obviously they have to get NIH compliance criteria such as IACUC approvals, um, IRB approvals, or IBC approvals. And third year funding is dependent on having that R01 level proposal submitted. And success is receipt of that funding, and when that happens, the funding starts, then that junior investigator rolls off, and that slot opens up to a new candidate. And total RPL funding cannot exceed 3 years, and that's from any COBRE source, so not just this center. And of particular interest for those at UNMC, there is going to be a junior investigator hire at UNMC, and that search will begin next year. And the hire would start at the beginning of year 4 with a maximum of 2 years of funding. That will be a tenure track position, and it's similar to an RPL in that they will have $200,000 a year in funding from the COBRE, excuse me, and they will have matching funds for those 2 years from UNMC. And that particular focus of that successful hire will be mechanisms of hearing loss during central neurodegenerative disease. So that could be aging or Alzheimer's or any other central neurodegenerative disease, so long as it was focused on hearing and or vestibular deficits. They would have an internal mentor, and the search committee would be led by Anna Junevsky, the director for UNMC, and the search committee would interview the candidates, and they would provide a short list of preferred candidates to the external advisory committee for the center. And I can talk about more of those much later, but they oversee the progress that we make within the center. And this particular hire is expected to be in either neurological sciences or in pharmacology and experimental neuroscience. And they would prepare a research project leader proposal or R01 level proposal. And funding from the COBRE would end when either the startup funding would end when if they got an RPL slot or they got R01 level funding. And but so that's for faculty level positions, but we also have funding sources for other projects to recruit individuals to the bench of, of faculty who could then for research project leaders. And these are funds for pilot projects to acquire the data to make a competitive RPL proposal. Again, these meritorious proposals will have a focus aligned with the center's mission. That is mechanisms of hearing loss or strategies that preserve, restore hearing. And there are about 4 or 5 of these pilot projects that will run for 1 year, each at $50,000 per year. And applicants are, at the moment, have to be eligible for an RPL position. Thus, they must be on a 10-year track. They haven't had an RPL slot before, and they obviously submit a meritorious proposal. And they must have a 10-year track position at any of the 3 institutions, Creighton, Boys Town, or UNMC. They will propose an internal mentor, and the mentor will provide feedback, and proposals are reviewed internally, and then that review is overseen by the external advisory committee. The review will have NIH benchmarks, so not only an impact statement, but also a significance, investigators, innovation, and the approach for that particular project, and those will all be scored. Um, there is a potential for second year funding via competitive review, and it prepares the applicant to submit for RPL proposals. And again, we're trying to get our trainees into the mindset of writing competitive proposals for NIH funding, because that's what's needed very much for an academic research career. Senior faculty may soon become eligible, but only if they propose a new direction from one that they're already doing. And everyone must have typical NIH compliance criteria, in other words, ICAR. We also have research vouchers, again, in a focus aligned to the center's mission. These are available in $1,000 increments up to a max of $5,000 per cycle. They're going to be about 2 cycles per year, and it's to acquire data to be competitive for a pilot project award. Currently, we want them to be only individuals who have a tenure track position. They're reviewed internally. And the most important thing here is that the vouchers are to be spent in the center's course. We have 2 cores, and I'm going to introduce those to you at this point. So we do have a question here from Dong Wang. Is research track faculty eligible for the COBRE pilot grant? So if they are eligible for the tenure track, then certainly. It depends. I don't know UNMC policy sufficiently well. Is the research track also a tenure track?
No, it is not.
Can anyone say?
Yeah, this is Chi-Chi. It is not a tenure leading track. Can you hear me?
Yeah, it's
Yeah,
not.
it is not a tenure leading track.
I can hear you. Thanks, Chi-Chi.
Mhm.
Right. So if by the time the award was made that the institution could have them that the applicant be on the tenure track, then that will be okay. So because it's a $50,000 award, so that is something that we can continue to discuss between the institutions to make sure that, that's possible. I have been strongly encouraged to ensure that pilot awardees are on the tenure track before making the award. So good question. Thank you. So the research cores, we have 2 research cores, the drug development and delivery core, and this is being led by Jian Zhou. And we have 2 co-directors, Shandor Lovash, who is very much an expert in both mass spectroscopy and protein structure modeling and bioinformatics. And also Alika Dash, who is a professor in pharmaceutical sciences and is very much a lot of expertise in pharmaceutical formulations. And the laboratory is managed by Gopal Jadhav, who is a medicinal chemist. And all of the services that we'll provide that I'll describe are provided not only to center investigators, RPLs, pilots, but also to outside investigators and companies on a fee-for-service basis. And there are 3 or 4 main applications of techniques in this core. The first is to develop in silico, in vitro, and in vivo high throughput screening paradigms to discover and validate novel therapeutics. So not only the in silico, but we can also use zebrafish screens or explants of mammalian cochlea or cell lines to identify candidate otoprotectants against different forms of acquired hearing loss. And these can identify many FDA-approved drugs that can then be repurposed. And they also have a medicinal chemistry pipeline to optimize chemical entities that are hits, but may not have um, ideal criteria or therapeutic indices because the dose is too high or the toxicity is too low. So they would make derivatives of those chemical entities to increase that chemical or therapeutic index. And so they have utilized existing medicinal chemistry facilities to optimize multiple drugs and synthesize new drugs such as Protax that involve ubiquitinases to digest endogenously synthesize proteins so that the absence of that particular protein causes otoprotection. And they are going to develop drug delivery and pharmacokinetic and pharmacodynamic methodologies using UPLC mass spec analysis of perilymph derived from guinea pigs, or for developing new techniques using MALDI mass spec imaging so that we can determine the cellular distribution of drugs within the cochlear tissues. And then the fee structure is to provide for acute sustainability for all these DDD services. And a lot of the links that we have on our website, I'm going to provide to the community in a PDF that will be distributed after this seminar, and I will provide these slides as well in a PDF. And then we've had some success in mentoring and training of trainees, like an MD PhD student, Jonathan Flegel, for an F30, and for a postdoc training award with Matthew Ingersoll, and also an early career stage researcher in Santa Nuhati. And Jay-Z, or Dr. Zoe, he is also one of the founders of Ting Therapeutics that has now received an SBIR Phase 2 award from NIDCD to further optimize his particular compounds. And so we have this increasing knowledge base that will help all of those that want to use these facilities. The other research core is the Auditory and Vestibular Technology core, or the AVT core, and again it will provide these services for all users. Obviously, RPLs, pilots, and center investigators are the preferred users, but as time allows, others will also be able to use them. And one of the major state-of-the-art methodologies they provide are both cellular and non-invasive electrophysiological and neurophysiological services of the auditory and vestibular system, and particularly non-invasive electrophysiology. So this includes things like auditory brainstem responses, distortion product, otoacoustic emissions that are very specific to these particular sensory modalities, and these will be available to researchers from any institution to have access to. And then we also have confocal imaging, molecular biology, including single-cell RNA-seq, and additional mass spectrometry services. And really important here for those at UNMC is that the ABR and DPOAE non-invasive electrophysiology services are available at the Durham Behavioral Core that's being led by Mysteria Samuelson and set up by Padmasree Rakanathan. So this will avoid the need to transfer mice or other rodents from UNMC to Creighton because this facility is now being hosted in the Durham Research Centre. And the same is also true on a more long-standing basis at Boys Town under the guidance of David He and Dominic Cosgrove. And they will also mentor core users in multidisciplinary research to successfully compete for research funding. And one of the ways they've particularly done this is in a RO1 research proposal that was scored highly and is potentially fundable. So we have a lot of training expertise and mentorship to enable people to succeed. And they have also developed a sustainability plan and a fee structure, and I'll provide those after the seminar. And one of the other things that the center is doing is hosting an annual Bellucci Symposium. And this year, uh, we give a prize as well, and the keynote address was given by the 2021 prize winner, Lisa Cunningham. And we made this award to her before it was known that she was going to become the intramural director of NIDCD. So so she earned this on merit. And we're going to have another one-day symposium early June of next year, and that will have a focus on age-related hearing loss. So we will distribute this information closer to the time once we have our speaker list assembled. So the innovation of this center is a thematic one. It's developing valid therapeutics that are efficacious in medical settings in which they are employed. And we have a mentorship plan that not only includes internal mentors, but external mentors to guide that translation into human studies. And the External Advisory Committee will review the proposals and overall center performance. We also have a larger sustainability plan, which is that Creighton, Boys Town, and UNMC are all putting in resources to recruit eligible faculty hires for the bench of replacement RPLs. The interactions with local COBREs and IDEA funded awards have increased our ability to be more effective. And in doing that, so we can replace COBRE funding with other funding, other support to support the center. And this includes independent research funding, industry, licensing, patents, and hopefully even sales or royalties. And SharpHub, which is another resource, I think, funded by NIH and I think that will help people guide the translation of their research data into commercial spin-offs. And so we apply for patents. We want to establish an incubator here at Creighton, and we want to attract sponsored research contracts. And we have had contracts from Novartis and Boehringer, Decibel, Frequency, and Manros. And we want to build a revenue stream for longer term stability. So I'd just like to close on some comments. So funding opportunities exist for UNMC investigators within the center's theme. And we have found that this infrastructure, in order to where we need to write proposals and manuscripts for the COBRE, forges closer interactions and collaborations, not only between investigators, but also within institutions. And it encourages us as the leadership of these centers to challenge the institutional administrations to act. They can be notoriously slow, but we use the deadlines in a manner to benefit us by forcing the institutions to act. If they say no, well, they probably would have said no anyway. But if they say yes, then we move faster. And so all of this enables research growth and translation into the clinic to impact lives. And the themes and the funds and the oversight by the EAC challenges our investigators to remain focused on our center's mission. And we can redirect resources to new specialized topics of actual need. And I couldn't have put this center together or even this presentation together without all of these individuals. So I won't name them all by name, but there is an executive team, the external advisory committee. We also have an administrative advisory committee and an internal advisory committee that includes institutional officials from both Boys Town and UNMC, and then Creighton officials for having the courage to support this venture. And so with that, I would like to thank you all for listening and to answer any questions that we may have.
Questions? Well, I think this was very, very informative, Peter, and we thank you very much for giving this talk today, because I think the number one thing is we have a much better understanding of, you know, what direction you're going, what resources you have, and kind of what constitutes your center for your COBRA. So this is good information for us to have. Does anybody have any questions? Final questions for Dr. Steyger?
Well, that's okay. I'll be happy to answer questions by email. That will be available in some of the materials that I share afterwards. And individuals from the center have also been presenting at UNMC, such as the Drug Discovery Corps. And so we all have many talents and different expertise, and we can all help one another in different ways. And so once we can identify how we can help each other, we can really have a synergistic impact rather than continuing to go at this alone without any additional input. So we're ready to help, and I'm pretty sure there's many of you out there who can help us. So thank you very much for inviting me, and I hope to talk to some of you soon.
Okay, thank you. And be sure and send us your PDF, and Heather will make sure we get it posted with your presentation.
Okay, great. Thank you, Chi-Chi.
Thank
Have
you.
a great afternoon, everyone. Bye.
Everybody have a nice holiday.
Yeah.
Great, thank you.